In the last video, we talked about mismatch of p orbitals of different sizes and how it's difficult to form an effective double bond or double pi bond under those conditions. And we showed some elementary examples of consequences of that. Mismatch of p orbitals of different sizes has very important consequences in biological systems. And in this video and in the next video, we want to show two demonstrations of how mismatch size of p orbitals is very relevant to biochemistry. Now, if you remember back in, I think it was videos number 14 and 15, we talked about nitric acid. And, by the way, there was, going back to even before then, starting at video 4 and going through video 19, we discussed different types of resonance systems. All of them were non-aromatic. And after video 19, we had several videos that dealt with aromaticity. But we're now going to be building upon the background that we established in these earlier videos. If you haven't watched them, I think you'll find them worth your while. Resonance is a very important concept in organic chemistry, and we tried to show many examples of different types of resonance systems. Now, remember back in video 14, when we first started talking about nitric acid, this is the structure. It has that covalent bond here between nitrogen and oxygen. Now, in the periodic table, directly beneath nitrogen is phosphorus. So, what would happen if we substituted phosphorus for nitrogen? Like this. And, well, let's go back to nitrogen for a moment. This double bond between nitrogen and oxygen is because there's a pi bond, an overlapping of the p orbitals between oxygen and nitrogen. Again, we discussed that in more detail, how the oxygen is sp2 hybridized. And when you have this overlapping of p orbitals, that's how we form our typical double bond or the pi bond. Now, if we have phosphorus in here, this pi bond does not form. And the reason is oxygen, that has a 2p orbital. Phosphorus is 3p. You don't get the overlap nearly as well, therefore the bond is extremely weak. So therefore, phosphoric acid does not exist in this form. Instead, you will see single bonds. So phosphoric acid has this structure. Like this. And the reason for it is, the reason for having these single bonds is because the pi bond does not happen because of the ineffective mismatch here of the 2p, 3p orbitals. You don't get that overlapping of them as effectively as if you have, say, nitrogen and oxygen, where the p orbitals are all the same size. So we're not going to see this for phosphorus. Instead, we see this. Now, in books, you will see phosphoric acid written with a double bond. But what happens is this. Remember, these hydroxyl oxygens, they have an sp2 lone electron pair and a pi electron pair. We're just going to draw in the pi electrons. And again, the fact that this is sp2 hybridized and has an sp2 unshared pair of electrons as well as a pi electrons unshared. We've covered that in previous videos. Now, for phosphorus, its orbital diagram looks just like nitrogen in this regard. You have an s orbital and the 3 p orbitals. The s orbital has a pair of electrons. The p orbitals each have a single electron. But unlike nitrogen, this is now the 3s level, and these are the 3p orbitals, not the 2p ones. Phosphorus also can expand into the d orbitals. So what can happen here is these pi electrons can go ahead and fill an empty d orbital for phosphorus. So these can come down like this and fill that empty d orbital. So we have this kind of a situation. There's another canonical form then that we can draw for phosphoric acid, and that would be like this. Oxygen has a positive charge because it donated both of its lone electron pairs to fill that empty d orbital in phosphorus. But it didn't have to be this oxygen that donates the electron pair. It could have been this oxygen. So another way that we can write the canonical structure for phosphoric acid would be like this. There is another canonical structure that we can write for phosphoric acid, or it could be that this oxygen donates its electron pair to the empty phosphorus d orbital. Can we write that here? So we have this. Like this. So for phosphoric acid, there's 1, 2, 3, 4 different canonical structures. But again, we have to emphasize the double bond that you're seeing does not result from p orbital overlap. The double bond occurs because oxygen is donating its electron pair to an empty d orbital in phosphorus. And of course it could be either this oxygen that does that, this one, or this one, so we can write these different canonical structures for phosphoric acid. And again, as we pointed out in previous videos, the more canonical structures you can write, the more stable a molecule is. But again, to emphasize, unlike the other videos now that you saw in the previous series, these different canonical forms that we're writing, these are not a result of p orbital electron delocalization. This is different ways or different oxygen atoms donating their pi electron pair to an empty phosphorus d orbital. But again, the fact that we can write these different canonical structures adds some stability to the system. Now, it's not a real good form because oxygen is fairly electronegative and it has that positive charge on it. Now, suppose though that phosphoric acid loses some of its protons. You will see phosphoric acid with a double negative charge. Now, if you remember though, back from, I think it was video number 13, we talked about carbonic acid, where it can lose a proton to form this anion, and that anion has a pretty decent resonance stabilization. But now, if this anion loses a proton to form this anion with a double charge, this has a very strong resonance system. Yet this hardly happens. And the reason is you've got this anion with a double negative charge. When the proton tries to separate it, it's going to pull it right back in. So this is not very likely to happen, even though this is strongly resonance stabilized. And again, we talked about that in detail in video number 13. Well, if it's difficult to form this doubly charged anion here, why is it possible to do it with phosphoric acid? And the reason most likely is this. This is a much smaller compound than phosphoric acid. Carbon and oxygen are much smaller molecules than phosphorus is. So here you have that double negative charge crammed into a very small volume, so that's going to be a very concentrated negative charge pulling back on this proton, making this very difficult to happen. Here, when you have phosphate, this is a much larger anion. The double negative charge is not quite so concentrated, making it possible then to lose 2 protons from the phosphoric acid. Now, when that happens, again, any one of these oxygens can donate its electron pair to the empty d orbital. Then we have these canonical structures. And notice these are better than the ones that we just wrote previously for 2 reasons. Oxygen has a negative charge instead of a positive charge, and there is no symmetry with these different canonical forms. So, what we want to establish here is phosphorus is acidic because you can write better canonical structures when you get rid of actually both protons. But this is not a typical resonance system now. These different Lewis structures, they're not the result of p-orbital electron delocalization, as we talked about during the previous 15 or 16 videos. This is just different oxygens donating an electron pair to an empty d orbital of phosphorus. Now, something else we want to discuss. Here is 2 carboxylic acids. They can lose a water molecule and hook together like this. This is an acid anhydride. Now, is it possible to form an acid anhydride, say with nitric acid? So we'd have this. that would be an acid anhydride for nitric acid. But this can never form. And the reason is that the nitrate anion, as we talked about in video number 15, the nitrate anion has a very, very powerful resonance, making it very, very stable. So in aqueous media, this can never exist. It's going to get pulled apart immediately to form two nitrate anions, so that those anions can experience this powerful resonance stabilization. So an anhydride of nitric acid is not going to be found in aqueous media. What about then, say, for phosphoric acid? Can we have anhydrides of phosphoric acid? And the answer is yes. So this is possible to form. This anhydride. And the reason is that, as you saw, the phosphates, you can write those different canonical forms for them, giving them stabilization, but that's not nearly as powerful, that stabilizing effect. That is not nearly as powerful as what occurs when you have this very powerful resonance stabilization of the nitrate anion. What we're saying is that this, while it does provide a stabilizing effect, this, remember, is just different oxygens donating a lone electron pair to an empty p orbital. These canonical structures, they do not nearly provide the amount of stabilization that this resonance, this very powerful resonance systems does for the nitrate anion, where now you're having the delocalization of the pi electrons. This is very powerful, making this impossible to form in aqueous media. But in aqueous media, it's possible to make this because this stabilization is not nearly as powerful as what would exist. This stabilization for a phosphoric anion is not nearly as powerful as for the nitrate anion. Therefore, it's possible to make this anhydride, or this is called pyrophosphate. Yet here, there's a problem. If we look at this oxygen could donate an electron pair to an empty d orbital. So could this one. What about this oxygen? Well, it's sort of torn between these two phosphate groups. So it's unable to do so. So that means then that the canonical structures we could write for this, well, we're not going to be able to write as many of them as what we did for the phosphate anion when we wrote these, because this oxygen is unable to participate. So when we have this in water, its tendency is going to be to form two phosphate anions, which in turn are stabilized like this. So what all this means is that it's possible to form this in aqueous media, impossible to form this. This can now go into two phosphate anions. Furthermore, it's going to be an exergonic reaction because these we can write those different Lewis structures for, as we just demonstrated. Here you can't because this oxygen is torn, so to speak, between these two phosphate groups. Meaning then that since this is an exergonic reaction, this then is a high energy compound. And that is why then in biological systems you see phosphate is used to form high energy compounds. Usually you think of it in form of ATP, but this is the chemical concept or the chemical principle that makes the whole thing work. And again, it's all predicated upon, unlike nitric acid, phosphoric acid does not have this pi bond, and therefore it does not have, when you form the anions, phosphoric acid does not have this super powerful resonance. Therefore, it is possible to form the anhydride, the pyrophosphate, but because there is some stabilization of the phosphate anion like this, then this anhydride that is possible to form, its tendency is to form two of these, making this an exergonic reaction, making then phosphate a high energy compound that can exist in nature. And that's why phosphate is used instead of nitrogen, because of the fact that you don't form these the double pi bond. Here you have it, here you do not. So the whole thing is predicated upon the mismatch of the p-orbitals of different sizes, and there is a demonstration then of how that is very important in biological systems.